Voilà, c'est lancé.
Bonjour, Monsieur le Président, Monsieur le Secrétaire perpétuel, professeurs, Mesdames, Messieurs, bonjour à tous. Merci de nous rejoindre pour ce qui sera notre 13e et en principe dernière séance en ligne. Dès le mois prochain, nous lancerons la formule hybride, en partie dans la salle Roi-Baudouin, en partie sur Internet, afin de respecter les règles sanitaires en vigueur et également de permettre à chacun de se déplacer s'il le souhaite. Aujourd'hui, et comme toujours, vous aurez l'occasion de poser vos questions à nos orateurs. Pour les membres de la RMB qui nous suivent, pas de changement. Le principe reste le même. Vous pouvez poser vos questions dans l'onglet questions-réponses ou Q&A si vous avez la version anglaise et nous vous donnerons la parole en live si nous avons un petit peu de temps pour le faire. Petit rappel de ce que vous allez voir s'afficher sur votre écran. En principe, vous devriez voir la slide apparaître. Vous allez avoir l'impression qu'on vous déconnecte. Cela ne va durer qu'une fraction de seconde. Pour les spectateurs qui nous suivent sur YouTube, vos questions nous intéressent également, bien sûr. Merci d'utiliser le chat dédié et d'indiquer dans tous les cas sur Zoom et sur YouTube votre nom et prénom ainsi que votre affiliation pour que nous puissions vous citer. Le symposium d'aujourd'hui sera consacré à la Network Medicine, un terme apparu pour la première fois dans une publication scientifique en 2007, une approche qui a permis de révolutionner l'étude de certaines maladies. C'est le professeur Jean-Luc Baligan, comme vient de vous le dire le secrétaire perpétuel, membre de la première section, qui a préparé ce programme, on le lui doit, ainsi que l'organisation de cette conférence. Monsieur le Président, je me tourne vers vous. Georges Casimir, le premier intervenant, est un membre étranger de l'Académie. Il a accepté de se lever à l'aube pour être avec nous.
Oui, merci Vanessa. Donc, chers collègues, mesdames, messieurs, c'est un très grand plaisir pour moi également de vous accueillir aujourd'hui pour cette séance qui va être fascinante à propos d'un sujet d'actualité remarquable et dont le futur est certainement particulièrement important, Network Medicine, la médecine des systèmes. Alors, c'est clair que l'ensemble de nos orateurs aujourd'hui vont s'exprimer en anglais et nous allons donc poursuivre cette séance, bien entendu, dans la langue de Shakespeare. In recent years, remarkable progress has been made in the process of approaching more and more in depth the complexity of cellular functioning, descending more and more intimately into molecular mechanisms. A genetic individualization has naturally been associated with this process, which leads more and more to an individualized medicine, which is distinguished by specific modes of diagnosis and treatment. New biomarker and statistical analysis based on artificial intelligence are at the heart of this work. More than ever, cooperation between various professions in medicine is essential to the process. Today, we are going to discuss this fascinating aspect. The symposium is organized by two sections of our academy under the direction of Professor Jean-Luc Baligan, chairman of the first section. Our first speaker is Professor Joseph LOSCALZO. Professor LOSCALZO is member of our academy, and we are particularly happy to receive him today, even if the health crisis forces us to do it again by video conference. He is Hersey Professor of the Theory and Practice in Medicine of Harvard Medical School, Chairman of the Department of Medicine, Physician-in-Chief, Symposium Distinguished Chair in Medicine, Brigham and Women's Hospital in Boston, Massachusetts. He trained in internal medicine and cardiology at Brigham and Women's Hospital, after which he was appointed to the hospital staff and Harvard Medical School faculty. After 10 years on the Harvard faculty, Dr. LOSCALZO moved to Boston University as director of the Whitaker Cardiovascular Institute and chief of cardiology. In July 2005, he returned to the Harvard faculty in his current role. author of more than 1,000 articles, 50 books, and 32 patents, is internationally recognized for his work on the vascular biology of nitric oxide, redox biology, systems pathobiology, and network medicine. He has received many awards, including election to the American Society for Clinical Investigation, the Association of American Physicians, the American Association for the Advancement of Science, National Academy of Medicine, the American Academy of Art and Science, and our Académie, Académie royale de Médecine de Belgique. He has served on several NIH study section and editorial boards, including the New England Journal of Medicine for 9 years, editor-in-chief of Circulation for 12 years, and is currently editor-at-large for the New England Journal of Medicine and senior editor of Harrison's Principles of Internal Medicine. Professor LOSCALZO will talk about the next medicine, novel approach to disease and therapeutics. Professor LOSCALZO, it's yours.
Thank you very much, Professor Casimir, for that very kind introduction. Are you able to see the screen?
Yes, we are.
Great. Well, thank you very much for kindly inviting me to participate in this wonderful symposium. I'm delighted and honored to do so. This is an exciting time in this new and evolving field of network medicine, and I'll try to share that excitement with you over the next 25 minutes. Here are my disclosures. So the first question is, why do we need a new approach to disease? And I would answer that question by arguing that conventional reductionism provides increasingly limited insight into understanding biological complexity and complex disease phenotypes. So we need a new approach, given how much more information there is available about disease mechanism and complex therapeutics. Now, there is a long arc of reductionism in biomedicine. I would be remiss if I didn't mention Descartes at the top of this list, but as applied to medicine, a good example of this reductionism arc is illustrated in the panel on the left from a paper my colleague Jeremy Green and I wrote about 4 years ago. In this paper, we talk specifically about the pattern of investigation in disease mechanisms over the last 200 years. And we use as an example tuberculosis, beginning with the notion of the sick person with phthisis in the 18th century, moving through to identification of the organs and tissues involved with more precision visually using histopathology. Then in the late 19th century, identification of the microbe responsible for the disease, and then ultimately identifying patterns of therapeutic strategies that have led to an increasing level of control of the disease. But the problem, of course, is that even before the anti-tuberculous therapies were identified, the disease was changing its ecology, and that led to a reduction in the severity of illness and to its infectivity, really illustrating how complex this interaction between microbe and man is. So the 21st century challenge, in contrast to the reductionist challenges of the last 2 centuries, is the challenge of reassembly, of understanding disease holistically, integratively. I think we have William Osler shown on the right, among others, as responsible for this notion of one system, one organ, one disease, one mechanism. And while that has held us in good stead for many years, it certainly has now run its course as disease definition becomes more complex itself and mechanisms become more difficult to dissect. Now, this problem of reductionism continues right through to the current century with the modern genome. And analyzing the genome as we typically do, illustrated here with a Manhattan plot for Crohn's disease, really is an exercise, again, in association and reductionism. As you can see along the 23 chromosomes, there are hotspots or sites shown in green that statistically significantly associate with the disease. This linear narrative of disease is helpful for us in identifying very strong and potent determinants of disease, but yet there aren't sufficient mechanisms or data for dissecting interactions or complex relationships, which can be much more informative in identifying therapeutic strategies and in determining the optimal approach for diagnosis. The importance of genomic context is critical here. And as this summary of a paper published now in 2016 by Steve Friend's group illustrates, even in highly penetrant, conventionally Mendelian diseases like Smith-Lemli-Opitz disease or epidermolysis bullosa simplex, that even in this setting, if you evaluate a number of patients, you'll find individuals who do not have any clinical manifestations of the disease. Here they screened 874 genes and found 13 adults with 8 mutations for severe Mendelian conditions, which they should have manifest, but did not. And their conclusion was these, quote, individuals are buffering the effects of rare, highly penetrant deleterious mutations, unquote. Well, that's really a tautology. The real question is how. So that kind of challenge led us, and in this case, the us is Laszlo Barabasi, a physicist and network scientist, and I, along with my colleague Ed Silverman, to help develop this field of network medicine, which is really a complex systems biology and network science approach to disease and therapeutics. It's predicated on the notion that most biological over the last decade, there have been new quantitative approaches that can be used to analyze these networks. This field, I shamelessly show a copy of our book from published about 3 years ago that highlight summary features of the discipline. And even in our Department of Medicine, we have a division that focuses on network strategies for diagnosis and therapeutics. So the first key question is, can molecular networks define genomic context and give unique insight into disease pathogenesis? And as an hypothesis that was tested, Barabasi and I published in 2011 a concept paper that made the following claim, that if one looks at an integrative network of proteins that associate with each other within the cell, within that interactome, that protein-protein interaction network, one can find discrete subnetworks comprised of a more limited number of proteins that are responsible for a specific disease, which is to say, in this universe of protein-protein interactions, disease-determining proteins comprise discrete modules, disease modules, as we call them. And the basis for this hypothesis, the bases are listed here, that proteins involved in the same disease tend to interact, that proteins involved with the same disease therefore tend to cluster in connected subnetworks, that proteins in a disease module are often involved in the same biological process, and these processes are discrete and separate from other proteins involved in other diseases, unless there is a common mechanism between 2 or more diseases. So these logically follow from the comments I made on the last slide. And then 4 years later, we published a paper that really provides the evidence for this notion of discrete disease modules in this comprehensive protein-protein interaction network. And on the left, you can see in the whitish background, the actual interactome that we used in this study. It was comprised of over 13,000 proteins that associate with, on average, between 10 and 11 other proteins. That accounts for the 140,000 edges, each of which reflects a discrete interaction between two proteins. And with this interactome, we asked the question, among the 299 diseases that had a sufficient number of known genes or proteins associated with them, do those disease proteins cluster into discrete modules? And I show, illustrated here schematically, 3 different diseases or groups of diseases that do so cluster. And notice that rheumatoid arthritis and multiple sclerosis overlap somewhat, which one would expect given their immunologic underpinnings. So how does one create a disease module? How do you derive it from the information in the experimental literature? Well, here's an example of such a module's creation by Rusheng Wang in my group. We chose, in this case, for illustrative purposes, cerebrovascular disease. We ascertained the disease network components or seed proteins, shown in blue, that associate or are known to associate with the disease. We map the seed proteins to the interactome, and then we identify connector proteins, shown in gray, which weren't previously known to associate with the disease. And then we statistically assess how discrete and separate this module is from another module comprised of a random number of proteins in the interactome. So the key message in this slide is that the interactome provides the missing links among the disease-associated proteins in the disease module. That gives the module integrity, and it begins to identify pathways that regulate the disease that may not previously have been recognized. Now, I don't have time to go through these specific examples in any detail, but here I illustrate 6 diseases that we have examined in this way, we and other groups have examined in this way. And from this examination, we've identified novel disease mechanisms or drivers that weren't previously appreciated. And as you can see, these disorders run the gamut of diseases from preeclampsia to aortic valvar calcification, to steroid-resistant asthma, to type 2 diabetes, and more recently to pulmonary arterial hypertension and perivascular fibrosis, as well as hypertrophic cardiomyopathy. And I'll come back to a couple of these in a moment. So now, can we bring together this notion of an interaction network and the conventional genomic notion of genetic variants associating with disease? How does one inform the other, or does one inform the other? Well, in a recent paper earlier this year, my colleague Fei-Sheng Chung and I, and a host of other collaborators, published a paper which I think begins to make the argument that network analysis does indeed inform genetic variation associated with disease and vice versa. In this case, we looked specifically at mutations shown in the yellow stars that are found on proteins associated with a particular disease, and their localization to the interface binding site between 2 proteins versus non-interface sites shown in blue. And an analysis, in this case, of germline mutations associated with non-malignant disease, an analysis of the localization of these mutations. Obviously, we had to know the 3-dimensional structures of the proteins to do this, indicates that there's between a 6- and 8-fold increased prevalence of mutations at interface sites versus non-interface sites among mutations known to cause germline disease. So that was pretty exciting. Fei-Sheng went on to look at somatic mutations in cancers and found a similar relationship, about an 8-fold difference in the prevalence of mutations at interface protein binding sites compared to non-binding sites for all but one of the malignancies that he explored in the Cancer Genome Atlas. So I think these are fairly convincing data of the importance of protein-protein interactions in governing disease mechanism. So now, if we know the relationship between genetic variants and network-associated proteins with regard to disease, can we begin to utilize this information to facilitate personalized genomic medicine? Well, to address that question, I'll give you an example of a recently published work on hypertrophic cardiomyopathy. As many of you know, this is a highly common genetic cardiovascular disease. It is associated with thickening of the ventricular myocardium, as illustrated on the right. There have been over 11 sarcomeric proteins with more than 1,400 genetic variants that are believed to associate with the disease. Yet, its clinical manifestations are of highly variable penetrance. Put another way, only about 25% of patients with hypertrophic cardiomyopathy actually have mutations or variants in the sarcomeric proteins that are believed to be major drivers of the disease. So clearly, the phenotype is much more complex than was originally believed. Now, to address this complexity, and this is a hard slide to follow, so you can use it for sort of background music, and I'll try to summarize it more succinctly. What we did was we obtained heart tissue from patients undergoing septal myomectomy, septal reduction surgery, to limit the aortic gradient. And we obtained from that septal tissue gene expression data, transcriptome data, and we compared that transcriptome to the transcriptome of myocardial tissue specimens from normal hearts that were about to be implanted in patients receiving a heart transplant. These hearts, as it were, ultimately rejected for implantation because of size differences, so we weren't harming heart tissue that would have gone into the patient. We then used a variety of statistical methods to look at differential gene expression and its localization, the location of these differentially expressed genes in the interactome. And this analysis really involved looking at the consequences of specific variants influencing or perturbing correlations across the entire gene set. And that kind of analysis, this pairwise perturbation correlation analysis, led us to be able to create an individual protein interaction network for HCM for each patient. And here we see at the bottom of the slide an example of such an interaction network, an HCM interaction network. We performed this analysis in 18 patients. We had 5 or 6 controls, and you're obviously not able to see anything more than these little fuzzy images here. The goal here is really to illustrate for you that even at this distance, one can appreciate the differences in the disease module for HCM for these 18 patients. And as we explored the specific differentially expressed genes in the different subgroups of these personalized interactomes, which I call reticulotypes after the Latin for network, is that certain subtypes of HCM have certain clusters of disease drivers or pathways in their disease module. And to be more specific, on the right, we illustrate examples of a subgroup of patients in these 18 who have enhanced fibrosis with a significant increase in the pathways that drive myocardial fibrosis represented in their reticulome or reticulotype. Using this kind of approach, we were able to identify a completely novel driver of HCM, which is JAK2. This is a well-known mediator of many malignancies, had not previously been recognized as important in cardiomyopathies or HCM in particular. And because there are available drugs that have been used against JAK2 associated malignancies, we have at hand the possibility for drug repurposing for this disease, which is the subject of ongoing work. So one can think of a network-based strategy in the following way. The genotype from genomic analysis of a patient, the genetic variants, can now be used to inform a reticulotype, which is a personalized disease network that can help distinguish among the different clinical manifestations of disease person to person. We can explore other related networks like endophenotype networks that govern intermediate phenotypes like inflammation, thrombosis, or fibrosis. And we also have developed similar strategies for physiological phenotyping networks, which combined allow one to begin to predict drivers of disease and clinical outcomes. So now in the last 10 minutes or so, let me talk a little bit about the answer to this question. Can this kind of protein-protein interaction network analysis facilitate drug target identification and guide drug repurposing as one strategy for drug development that's become quite intriguing of late? And the basic motivation for this question and this line of investigation is that since we now know increasingly the proteins that govern disease pathobiology, we are confronted with a much greater number of potential disease targets drug targets that one can use to develop specific therapies, at least theoretically. Now again, our approach to therapeutics is quite reductionist, and we have Ehrlich, shown here, to thank for this current strategy. His magic bullet theory represents another form of reductionism in practice. Remember that he was a dye chemist, and his notion of the side chain theory of immunospecificity grew out of observations that dyes stained different tissues differently. So that he argued that there were specific receptors, although he didn't actually call them receptors, side chains that interacted with the dyes that were tissue specific. And the leap here that he made was the concept that if that is the case, then perhaps these tissue specific side chains could also be used as a strategy for directing specific therapies. And of course, he was really the first chemical biologist, first medicinal chemist who used low throughput screens by pulling chemicals off his shelf and testing each randomly. And of course, his first success was the treatment of syphilis, screened in this way with an arsenical, preparation 606. Now, let's keep the thought in mind that this reductionism, this notion of a magic bullet approach to treatments has not yielded what it had in the early days of pharmacologic discovery, as illustrated here from data that Harold Schmidt shared with me. Notice the y-axis is a log scale. There is an increasing limitation of the conventional target-based approach to identifying effective drugs. I think all folks in the pharmaceutical industry would appreciate that point. Another interesting point to remember about drug development is that approved drugs really lack efficacy. They reach statistical significance required by the EMA and FDA in clinical trials. But as shown here, for the 10 highest grossing drugs, most profitable drugs in the US, they fail to help between 3 and 24 individuals. The green people here are those for whom the drugs are effective. So I think it's a powerful slide and image, which highlights how many people we fail to benefit with the use of increasingly expensive drugs. Another point to highlight, of which you may not be aware, is the fact that drugs are very dirty. That even though we believe, very promiscuous, even though we believe that a drug developed by a very effective structural biological therapeutic strategy in a major drug company is highly specific for the target of interest. It is not. And here in a paper by Chartier and colleagues published a couple of years ago, in which they used a bioinformatic approach to ask the question, how often does the so-called specific binding motif for a newly developed drug, how often does that motif recur throughout the proteome? And what they found, I plotted their data in this distribution, but the data are theirs from their study. What they found was that on average, a so-called specific drug of the 475 they analyzed had on average 32 targets. Notice this is a very long tail distribution, and most drugs have a significant number of targets. Very few have one or two targets. So the reason that this is not emphasized is that no regulatory agency asks the pharmaceutical industry for this kind of unbiased analysis, A. B, the methods for approaching this kind of unbiased analysis until recently have not been available. C, they would divert attention from the focus of the development process, which stems from identifying a specific target as required by the regulatory agencies. But I view this as useful and helpful information, not unhelpful information, because it highlights A, why there are so many off-target effects and unpredicted toxicities once drugs are given to large numbers of patients. And B, it gives us a basis for considering the repurposable use of many drugs. So how can you think about drug target identification from a network perspective? Well, on the left, we show a typical schema for a disease module within which there is a specific target, shown in blue, against which a drug is developed. And one can analyze the pathway within which that target sits in a screen for compounds that may be helpful. But on the right, we have a different strategy. This is the network-based drug repurposing strategy, the proximity hypothesis, in which we first consider a disease module shown in pale blue here, within which there is a drug target for the disease, but next to which, T2, there's another drug target for another disease. But it's close enough to the disease module that we ask, can that same drug target and the drugs that are used to impair it, can those drugs be repurposed for this disease? An example of the answer to that question is shown here. This is recent work by Jun-Sup Song and Jane Leopold, in which they created the vascular calcification endophenotype module, within which they found some drug targets that, against which there are known drugs like everolimus and temsirolimus, as well as pomalidomide, and showed in an in vitro assay here that a smooth muscle calcification, that these drugs could significantly impair calcification in this in vitro assay, and therefore they could be considered as potentially applicable to severe vascular calcification endophenotype patients. We have several other examples of repurposing, most notable among which is repurposing drugs for coronary heart disease, for pulmonary hypertension, and in the interest of time, I can't share those with you today, but I will show you some very recent data that's timely related to drug repurposing using network strategies for SARS-CoV-2. Now here, the strategy is a little more complicated because we have to deal with three different networks. The viral protein network, there are 27 proteins that SARS-CoV-2 makes, and they associate in various ways. And then a disease module network within the host cell, that is a group of proteins that associate with the viral proteins. And then a drug target disease module that is nearby the human covidome, as we call it, through which one perhaps might be able to identify repurposable drugs. So we first created the covidome, and the actual covidome is shown here on the left. This was derived from the 332 human proteins that Gordon had shown last year in Science, bind to, I believe, 26 of the 27 SARS-CoV-2 proteins. So there is a discrete disease module in the interactome that reflects the human proteins responsible for manifestations of COVID-19 infection. And in addition to the network proximity method, we developed two other methods with collaborators that include a network diffusion strategy and an AI-based strategy that Marinka Zitnik helped us design in this paper that we published earlier this year. And using this strategy, we came up with a rank order of this combined strategy, a rank order for drugs that might be usefully repurposed for COVID-19. We then collaborated with Rob Davey, who runs the National Emerging Infectious Diseases Laboratory here in Boston. And Rob then tested in a high throughput screen the lead compounds and was able to confirm our predictions very effectively. And on the left here, I give you an example of a high throughput assay. The pinkish stain reflects viral antigens. The blue stain represents the nuclei of the epithelial cells. And as you can see, remdesivir, which was a positive control, was quite effective at inhibiting viral replication. In these assays, we can look at replication as well as cytotoxicity, and that's an important consideration. What we found were three other drugs that were quite effective: emetine, procelaridin, which is a cardiac glycoside, and obatoclax, which was quite intriguing. This is a BCL2 inhibitor used for the treatment of non-small cell lung cancers and lymphomas. It's experimental. And here we showed in an in vivo assay using mice that overexpress human ACE2 that obatoclax in female and male mice was quite effective in suppressing the viral titers to barely detectable levels in lung tissue. So that, I should say that methodology really is quite effective. It improves our ability to predict positive drugs in the high throughput assay from 0.8%, which is what the typical yield is for random high throughput screens of hundreds of thousands of compounds, to 35%. So our network-based predictions are quite useful, reduce expense and time, and highlight potentially unique repurposable drugs. Now the question is, can we expand the universe of repurposable drugs? And this is where work with the Lawrence Livermore Laboratory and Felice Lightstone, who is the head of their structural biology unit, comes into play. We have a funding from the Department of Energy and the US Department of Energy and the American Heart Association to establish the Center for Accelerated Drug Discovery. And we use their near exascale high performance computing system to do what would otherwise take about 20 years of in silico modeling to do with conventional pharma-based in silico screening. And we are in the process of creating a huge matrix that involves about 2 million open access compounds and about 14,000 human proteins whose tertiary structures are either known or imputed from their congeners in other species. This matrix will allow us for the first time to identify in an unbiased way drug targets unrelated to the drugs to the diseases for which the drugs were developed, and may give us a more rational guide to drug repurposing opportunities. So I'll end with this slide, which highlights the summary of our approach, our network medicine-based approach to disease diagnosis and treatment, emphasizing the importance of this notion of reticulotyping and of drug repurposing and novel drug target identification. We should, in the end, have subclusters of patients among those very many who don't respond to treatments that I showed you in that short slide, that we can now guide more specific targeted therapies more effectively. So I'll acknowledge the folks whose names I mentioned throughout the talk. On the left, collaborators, and on the right, those folks who did the work in our lab and other labs over the last 10 years. So thank you very much for your attention, and thank you for the kind invitation to participate in this wonderful symposium today.
Thank you, Professor LOSCALZO. We will immediately have a Q&A, and I see someone applauding, one of your peers is applauding on Zoom. I already have someone asking a question, so I will ask Professor Miriam Knop to unmute herself and to display her video if she can. Good morning, Professor Knop.
Good morning and good night, Dr. LOSCALZO. I really enjoyed your talk. That was beautiful. My question was about whether you can give your views on how we should build such networks for diseases where the genetic variation really lies in non-coding regions. I myself work on type 1 and type 2 diabetes, and 90% of the genetic variation that is associated with disease risk lies in non-coding regions. I feel there is a big knowledge gap that needs to be filled before we can actually build precise networks for those diseases. But what's your view?
Yes, thank you for that question, Professor Knopp. One of our first papers on network associations with disease was in pulmonary hypertension, now about 10 years ago, in which we created two networks, one of which was the protein interaction network, and the other was a microRNA network. And we mapped this network of networks, we mapped the microRNAs to their targets in the disease module for pulmonary hypertension. And we're able to identify a novel microRNA that had not previously been shown to regulate disease phenotype. That was microRNA 21, which is known to affect many other phenotypes, but never in pulmonary hypertension. So this notion of creating networks of networks that are, some of which are regulatory transcriptomic networks, some of which are non-coding RNAs, like including microRNAs and long non-coding RNAs and circular RNAs. This is a growing area, and it is completely consistent with the notion of complex interactions. It's just on a different level. So we published a paper that reflected some of this work in diabetes with the late Amitabh Sharma last year. And in this paper, we looked specifically at, theoretically and through in vitro assays, at associations of non-coding RNAs to certain pathways that drive disease phenotype. And I think this allows one to explore this notion of effects at a distance within a disease module. So if you have a cluster of variants in a pathway that you know affects some biochemically accessible element or feature of the disease, and yet there is another differentially expressed set of proteins at a remote portion of the interactome, it could be that distance, that separation, is a consequence of a regulatory feature that hadn't been previously appreciated. So it plays right in very nicely into making a complex story even more complex.
Thank you.
Thank you, Professor Knop. Professor Baligan, you have a question. I will ask you to unmute yourself.
Yeah, I hope you can hear me. So that was brilliant, Joe. Thanks again for your lecture. I just want to come back to the slide where you highlighted the fact that any drug has much more than a few targets. And obviously that property and that observation can be used for drug repurposing. But on the other hand, you could use this information perhaps the other way around and consider that all known targets that have been identified for each drug that works in a specific disease may actually indicate nodes of that disease that hasn't been anticipated before. And so could that be used to enhance our understanding of pathophysiology of the disease?
Yeah, no, absolutely. I think in this case, what you're suggesting, and I completely support, is that drugs can be viewed as perturbants to a unique network for an individual patient. And there are other drivers of the disease in addition to the single protein against which the drug was first developed that the drug may also influence or affect. Maybe other members of the same class of kinases, for example, weakly, but still. So yes, I think you can use this pluripotent perturbation effect of most drugs as a way to explore other features of disease drivers. You can also use this strategy as a way to identify drug toxicities, of course. We've, in ongoing work that we haven't yet published, we've done that for long QT syndrome, and we're able to identify the disease module for long QT syndrome as a side effect, and show that it overlaps with many other pathways in cardiomyopathies modules. So there are different, depending upon your perspective, you can use this very interesting observation of Chartier as a very usefully.
Professor Casimir, you have a question. I see other question in the chat. So if, may I ask you, Professor LOSCALZO, if you are going to stay with us until the end of the session, or if you are going to leave?
No, I'll stay.
You will be there for the roundtable then. Yes. You confirm that. So we will ask the other questions during the roundtable. So do not hesitate to flag your other questions. But Professor Casimir, the floor is yours for this question, and then we will move.
Yes, thank you very much, Professor LOSCALZO, for this fascinating lecture. My question is, could we imagine in the future that patients could carry a smart card indicating the characteristic of an important part of their proteome of protein interaction upon admission to the hospital in order to define part of the treatments that will be provided to them?
Yes, wonderful question. Wonderful question. I think I absolutely believe that that's the case. And in fact, it may be carried as a hologram. My colleague Barbassi is also a sculptor. And he has he's just had a showing of work, not of his, but of others, of 3- and 2-dimensional versions of networks in Budapest, which will be going to Munich next. And some of the concepts shown there illustrate how this kind of imaging, virtual imaging, you can sort of walk through the network and identify those areas where there are variants and begin to explore your own network and what risks you may have as a consequence. Wonderful question. Thank you.
Thank you very much.
Thank you, Professor LOSCALZO. We'll see you later. Just the time for us to talk about the second conference. Donc, Professeur Casimir, la deuxième conférence va nous projeter dans l'univers de l'intelligence artificielle et du machine learning. Sur le plan thérapeutique, les données traitées sont source de beaucoup d'espoir et aussi de nouvelles applications cliniques. L'orateur que vous allez nous présenter est belge. Il s'agit du professeur Yvan SAEYS.
Yes, our next speaker is Yvan SAEYS, co-laureate of AstraZeneca Prize in 2020, data mining and modeling for biomedicine from the University of Ghent. Yvan SAEYS obtained his PhD in computer science from Ghent University in 2004, after stays abroad at the University of the Basque Country and the University Claude Bernard at Lyon. He returned to Belgium, where he founded the Data Mining and Modeling for Biomedicine Group at University of Ghent. Currently, he holds a professor position at the Department of Applied Mathematics, Computer Science and Statistics at Ghent University, and is a principal investigator in the VIB Center for Inflammation Research. The SAEYS lab is widely recognized for its expertise in machine learning methods for single-cell biology, mainly applied in the field of immunology. Research in the lab spans the whole spectrum of methods and technology development up until application in the clinic. The title of his lecture is Machine Learning, Clinical Application of Artificial Intelligence, Single-Cell Bioinformatics. Professor SAEYS, it's yours.
Thank you so much, Professor Casimir, for the nice introduction. I changed a little bit the title to make it more comprehensive. So in my lab, we are indeed using methods from the broad field of artificial intelligence to get more insights in data that we mainly obtain using single cell technologies. And we're using that for a wide variety of applications, mainly in immunology. And we're also trying to go one step further and actually translate then all these AI methods to the clinic so that they also become useful for the patients in the end. So let me just start by framing a little bit the broad research domain of artificial intelligence. So in artificial intelligence, we try to build systems that do things that we as humans would think require some form of intelligence. In order to do that, we mostly use engineering and mathematical approaches to build these kind of systems instead of really trying to simulate how exactly the brain works. And the key question within the field of artificial intelligence is actually how to find good representations of knowledge and of data that actually allow us to reason with these representations and do inference, right? So infer new knowledge from existing data and existing knowledge. And so the talk of Professor LOSCALZO was a very nice illustration of one type of representation, namely networks and graphs, and the powerful things you can do with such kind of representations. So let me just give a quick overview of the different subdomains in artificial intelligence. I'm not going to go into detail into all of these subdomains. I'm pretty sure you are familiar with many of them, like, for example, search and planning, robotics, computer vision, and so on. So these are all different subdomains. Of course, many of them hold great potential for applications in the health and clinical domains. But what I'm going to focus on today is one subdomain of artificial intelligence that we actually call machine learning. And machine learning is about models that we construct from data. So if you think about the classical way that a computer is programmed, you typically write lines of code where you explicitly say to a computer, do this, then next do that, and so on, to solve a certain problem. Now, in machine learning, we're going to apply another type of programming of a computer, not saying explicitly what the computer needs to do, but rather what we want the computer to learn by showing examples to the computer. So, for example, if you're interested in building a system that is able to recognize faces, you're going to show faces to the computer and tell them this is a face of this person, this is a face of that person, and so on. And by doing that many times, you would hope that the computer would actually learn from these examples a general representation of a face, for example. Of course, we can do very similar things for diseases, right? So we can represent to the computer a lot of different types of data, for example, that we collect from healthy patients and patients with a certain disease. And then the computer has to learn from these examples by using some kind of mathematical models that we typically use. We're not going to say explicitly to the computer what is the difference between the healthy and the diseased patients. The computer has to actually learn these differences just by us giving examples. And there are many types of machine learning methods that we can actually use. You can see them at the bottom of the slide here. So for example, one type of methods is about really learning structures in the data. So for example, if we have a set of patients having a certain disease, maybe there will be subtypes of these disease patients having maybe different mechanisms or different subtypes of the disease. And then you would want to learn this substructure that is present in your data. So those are typically called unsupervised models. Typical application are clustering techniques where we try to find subgroups, for example, of patients that are more related to each other within the subgroup than between the different subgroups. A second type of models are predictive models. So in these models, you try, for example, to learn a diagnostic model that is able to predict for a new patient if the patient has a certain disease. And this model is actually trained typically on historical data that you have been collecting in the past. A lot of these type of models are using what is now called deep learning models. So these are typically quite complex neural network type of models that have been shown to give very good performance, especially in imaging related problems. And then there's a last category of models that is about learning policies. So you can actually compare that with a child, for example, that has to learn to bike. As a parent, you're not going to say explicitly move your feet 5 centimeters up and down and so on. No, the child has to learn by itself, by falling and doing how to bike. And similar types of methods for learning such kind of control policies can also be done using machine learning methods. And they are typically used a lot in the robotics field, for example. So in my lab, we are using these machine learning methods to mainly analyze different types of single cell data. And the single cell field is moving very, very fast. So we are able now to profile in very detail different types of tissues of a patient. And of course, by doing that, we get unprecedented amounts of information that give us indications of really, the molecular workings of cells and tissues. And from that, of course, we hope to extract some mechanistic insights about how all these systems are working and how they're differing in healthy state and disease state. So of course, if you're looking for biomarkers, for example, for certain diseases, you're looking, for example, to easily available fluids, like, for example, blood. Apologies. And you can try to extract some features from that, for example, could be used in a diagnostic model. So the types of single cell data that we are working with, well, historically, we started with cytometry-based approaches because I'm mainly an immunology department. So cytometry is kind of a workhorse method for immunology. And so we used and we developed in the beginning a lot of methods to analyze flow cytometry data, but now we're moving to other types of single cell omics data where we can even measure the full transcriptome at single cell level. And now the latest technologies that we have is spatial omics technologies where you can even profile every cell at the single cell transcriptome level, but also find out the spatial location of all these transcripts. So that gives tremendous amounts of information. And of course, the challenge is then to develop novel models that can integrate these different types of data and of course get to novel insights. So what can we do with single cell data? Well, we can do a lot of interesting things like trying to find out the structure that is present, for example, the different cell types, how these cell types are related to each other, try to visualize that in a comprehensive way. We can get more mechanistic insights into the system's dynamics and the underlying regulatory networks that are driving cell developmental processes. We can go one step further and also try to find out how cells are communicating with each other, both in healthy state and in disease state. We can try to integrate all this data, for example, with clinical data and try to do patient stratification. And of course, if you collect all these types of data for a large amount of patients, that really leads quickly to a very large amount of data, and you need big data technologies to still make it feasible to process all these amounts of data. So a lot of our research in the last 5 years has been in really automating the analysis of single cell data, just going to show here a few examples on flow cytometry, where we actually developed a whole computational pipeline that consists of different steps, including automated quality control, normalization, automated cell type and identification, and then extracting properties from all these cell types that could be useful for predictive models that could be used either for diagnosis, prognosis, or extraction of biomarkers. And then, of course, in the last step, you need to validate all these models in independent external cohorts. So first example that we worked on was the automated diagnosis of common variable immunodeficiency, which is one of the most frequently diagnosed primary antibody deficiencies. The problem with the diagnosis is that it's typically a diagnosis by exclusion. So typically it takes quite a while before the patient is diagnosed with rightly with CVID. And so we try to set out if we could develop models based on artificial intelligence to, from a blood sample, predict if a patient has CVID or not. So this was work done by Annelies Emmanuel together with the lab of Philomeen Haring at the UZ of Ghent. And so we collected a quite substantial data set here, and then we applied this pipeline for computational flow cytometry. And what you see in these tables here is that with the manual analysis, the classical way of analyzing flow cytometry, you would reach about 80% accuracy of the models. But using this automated flow approach, we could push that over to 90% accuracy in diagnosing CVID versus non-CVID, and also CVID versus other types of primary antibody deficiencies and healthy patients. Of course, these models are interesting because you can dive into them and see which features the model is actually using to make the prediction. And from that, you can extract useful biomarkers, like here some types of B cells or T cells that came out of these models as potentially interesting biomarkers. The second application is on myelodysplastic syndrome. So this was a collaboration with some people from the Amsterdam UMC. So there we also try to automate the process of diagnosing these patients with myelodysplastic syndromes. Many of these patients have a high risk of progressing later to acute myeloid leukemia. And so we set up a kind of similar pipeline here. The idea was also trying to automate the process, but also make it a little bit faster, because the typical manual workup in the hospital requires about 1 hour per patient to analyze all the flow data and do the diagnosis. So we also made a completely automated system here. We trained it on a training cohort. We validated on an independent validation cohort. And you see that also here we obtain very high numbers for the predictive performance of the models that also largely surpass the manual analysis. What was even more interesting is that using these 6 tubes of antibodies that were typically used in the clinic, we could actually reduce that to 1 single tube of antibodies that has almost the same classification performance, but needs a lot less antibodies, so it's a lot more cost effective, and also needs thus less cells from the patients. So you have to take less bone marrow from the patients to have a classification model that has almost the same accuracy as the classical model. And very interestingly, also the time aspect of running the whole computational pipeline reduces the diagnostic process from 60 minutes, which would be the classical way of people doing it in the clinic, to 3 minutes or 30 seconds, depending on the computational method that we're using. So that's a huge saving in time. And for example, that would allow people in the clinic to maybe focus more their time on the more difficult cases. So also here we could extract some novel biomarkers from these computational models. I'm not going to go into detail, but some typical erythroid progenitor out, and they can be confirmed in the clinic as indeed being very important for the diagnosis here. So just to conclude my talk, I'm going to quickly give an overview of some of these single-cell RNA sequencing approaches. So in collaboration with the lab of Geert Bergs, we actually did a study on ovarian cancer, where we collected data from your ovarian tumor and also some sites of metastasis for a number of ovarian cancer patients. We're still a low number of patients here, 3 patients. And then we collected single-cell RNA sequencing before and after chemotherapy. And then the idea was to find out what is the heterogeneity of all the cancer and other cells that are present at these different sites of the cancer. And can we find, for example, typical marker genes for all these different populations? So you see here a UMAP plot, which is a 2D visualization of actually a very high dimensional data set, where you see the different clusters here representing the different cell types, so the cancer cells, the endothelial cells, and then different types of cancer-associated fibroblasts. And of course, all of them you can analyze more in detail because you have the full transcriptome profile. So at the right-hand side here, you can see which genes are specifically expressed then in these different subtypes. And of course, if you find these genes, you can actually use them later on as specific marker genes for example, specific populations that maybe can tell you something about chemo-resistant cells, and they could be used later on, for example, to design a novel flow cytometry panel so that you then can easily screen in a more cheap way these patients later in the clinic. I want to conclude by just a very short story about some novel methods that we have been developing on really trying to get more insights in the dynamics of intercellular communication, which actually nicely ties in with the network story by Professor LOSCALZO. So we're developing here some novel computational techniques that allow us to actually reconstruct how cells are communicating with each other. So for example, if you have a cell 1 here that through a ligand receptor interaction is talking to cell 2 here, that then this receptor sets into action a whole regulatory cascade here, you can actually look at the transcriptional footprint in this cell 2 here and try to predict what was the ligand receptor interaction between cell 1 and cell 2 that was going on. So in order to do that, we combine, again, using a network representation, different types of information like ligand receptor interactions, signaling interactions, but also gene regulatory interactions. And we combine that in a very large integrated network. And then we're using techniques like the Google PageRank algorithm to find actually influences of certain ligands to certain downstream target genes. And these downstream target genes can actually be seen as a footprint of maybe a potential ligand receptor interaction that is going on. So using this baseline network model here, we can actually combine that with a transcriptome of a single cell RNA sequencing experiment. And this would allow you to prioritize which ligand receptor interactions are actually active in the cell types that you're investigating. So this was published in a Nature Methods paper last year. And this is an example of this method in the liver. So in a nice collaboration with the lab of Martin Williams, we were looking at monocytes that are entering the liver and the niche signals that they get in the liver to develop into the macrophages of the liver, which are the Kupffer cells. And our long-term goal is actually to better understand how all these cells in the liver are working together to then later try to translate this into organoid models where we can actually develop in vitro liver mimics that, for example, would be very useful for all kinds of toxicity studies. So I would like to end by thanking all the people in my lab that contributed to all these different methods that we've been developing. And of course, also all the clinicians that we have been working with that were kindly enough to donate the data, but also to help in the interpretation of all the models. And I'd like to thank you for your attention and happy to take any questions.
Thank you, Yvan. Is there anyone? Yes, I see Professor MACQ raising his hand. So, Professor MACQ, the floor.
Thank you very much for the nice talk, Yvan. It was very interesting and very complementary to things that we do here. I had just a question. You started the presentation by speaking about different kinds of machine learning, and one of them is reinforcement learning. And my question is, in the different things that you are addressing today, do you already use reinforcement learning in your toolbox to solve problems for MDs?
No, we have not been using reinforcement learning so far. And also, if you look at the international literature, this is still kind of an underexplored area of machine learning, because typically it requires some kind of interaction with
a reward function that is difficult to design.
Yes. Yes.
Okay, thank you.
People have been mainly looking at the other types of machine learning, so the supervised and unsupervised methods where you actually only need data, right?
Okay, thank you very much.
So, Benoît, you also have a question.
Thank you. So, Yvan, you very nicely highlighted the logistic advantage and intellectual advantage of separating different cell types and characterizing the transcriptome on a single cell basis. And you use that to try and discern the different cell-cell communication pathways. My question is, obviously you could demonstrate some signatures of ligand receptor interactions that drive the expression of specific genes. But can you extend your model by going from the transcript to the actual protein and then to mediators that would be released in the intercellular milieu that would then feed back on other cells? And so in other words, do you think that the field is ripe now to go to single cell proteomics and try to move beyond the transcription regulatory network?
Yes, that's a very interesting question. Yes, all these other mediators that could be important for signaling between cells. So single cell proteomics is lagging a little bit behind with respect to what is currently possible for single cell transcriptomics. But people are working on that. So I've seen some first papers appearing last week, I think, from the group of Matthias Mann, who's very known in proteomics. So definitely people are working on that. And of course, that would be very interesting then to integrate with the transcriptomics data. There exists already some technologies to actually from the same cell measure transcript and protein information, but it's typically only surface protein like we typically do in immunology studies. But it will be certainly coming. Yeah.
Thank you.
And the last question will be for the president of the Academy, Professor Georges Casimir.
Thank you very much for your nice lecture. My question is there a need for special training today and a lack of biostatistician in the field of artificial intelligence and single cell data?
I think that's a very relevant question, because now that all these methods are being increasingly used also by clinicians, well, one often heard critique that we are also faced with many times is that people in the clinic say, well, we don't trust these methods because we don't understand how all these methods are working. Mm-hmm. So, of course, there is a need for training. We need to explain to these young scientists how all these methods are working. I think, maybe the role of the academy can be there to stimulate this more. but it's coming. I see all the young scientists, both from the clinical fields as well, they are also interested in learning how all these models are working. They're interested in analyzing their own data instead of sourcing it out to a bioinformatician. So I think things are slowly starting to change, but we can, of course, push that more and motivate them more.
Yes, it could be interesting to associate the universities to create new masters in this field.
Indeed, yes, thanks. And to also create novel research fields. I'm completely sure that will happen.
Thank you very much.
New projects for the Académie and for you, Yvan. Thank you so much. Aujourd'hui, l'état de conscience et les mystères du cerveau, eh bien, nous allons en parler. C'est ce dont va nous parler l'oratrice suivante. Monsieur le Président, là aussi, l'intelligence artificielle a permis à la science d'avancer et d'aider les patients et leurs familles.
Yes, of course. Our next speaker is Athena DEMERTZI, co-laureate of AstraZeneca Prize in 2020, and working at the GIGA Consciousness Lab in University of Liège. Dr. Athena DEMERTZI is FNRS Research Associate at the Institute GIGA since 2018. Holding a PhD in medical science in 2012 at Université de Liège, she has graduated as a psychologist from the Aristotle University of Thessaloniki, Greece in 2005, with a subsequent master specialization in cognitive neuroscience and neuropsychology at Maastricht University. In short, her research contributes to the knowledge about the human mind, even when this cannot be communicated overtly. Athena DEMERTZI currently directs the Physiology and Cognition Lab. With her team, they investigate brain-body interaction as a proxy to human sentience in health and disease by means of high and low-tech methodologies. She was recently awarded with the Scientific Award in Patient Care in the Artificial Intelligence IRE 2020 by the AstraZeneca Foundation for a contribution to individualized patient care by means of machine learning applications. She also received the FNRS Mandat d'Impulsion Scientifique for the project Evidencing Sentience in Low Arousal States by Probing Brain-Body Interaction. In the past, she was FNRS postdoctoral research and an INSERM fellow at the Institut du Cerveau de la Moelle Épinière à l'hôpital Pitié-Salpêtrière in Paris. Her PhD was funded by the DISCUS Marie Curie Research Training Network and the Université of Liège. The title of her lecture is Application of Artificial Intelligence to the Study of Consciousness, Neurology, and Neuroimaging. Professor DEMERTZI, it's yours.
Thank you so much for the kind invitation. It's a pleasure to be here today. Hopefully next time we can be in person. Yes, indeed, my background is for this lecture will come from the field of neurology, mainly. I've worked in the past, especially with brain-injured patients, where we have a very big problem that they cannot communicate to tell us how they feel, experience the world. And like that, we don't know their wishes for treatment options. So when I talk about disorders of consciousness, these are heavily brain-injured patients for whom we have to infer the cognitive capacities, their mind, from behavior. So in that case, people who find themselves in coma, they are considered unconscious because they cannot evidence it overtly, because even if they are severely stimulated, like even painfully, they will not open up their eyes and communicate. When we have preserved arousal levels, for example, by eyes opening, but patients are not able to communicate, they are supposed to be in a vegetative, unresponsive state. And they still remain and considered unconscious. We have sometimes some of these patients, though, that they can show some type of high order motor responses, like sometimes they will show non-reflexive movements, like following their image in a mirror, or they would respond to command, let's say a simple command like raise your thumb. And in that case, they're supposed to be minimally responsive in a minimally conscious state. And the problem still remains because we can still not communicate functionally with these patients. So in the absence of communication, how can we know that somebody entertains some subjective experience? How can we quantify their mind? My contribution to that comes from the paradigm of resting state. This is to say that when patients receive MRI scanning, it's very convenient for us because then we can ask the neuroradiologist to record functional data. So for 10 minutes acquisition using functional MRI, we are in possession of big amount of data that now we need to make sense of, like how this kind of functional communication between regions of brain regions can tell us something about the level of consciousness patients. So the resting state, indeed, it's not a condition where we do nothing. When we are even like not engaged in a task, when we stay silent, our mind is very, very busy. So we will entertain different types of thoughts. Our mind will go in the future, in the past. It will have a very rich mental repertoire. So this means that even that we have the evoked changes in the brain entertain less than 5% of the brain's energetic budget, because most of it goes for neural signaling. This means that conscious awareness is energetically inexpensive. But it depends upon a very complex and dynamically organized state of the brain, which achieves at great expense. Talking about networks, I will talk in my lecture mostly about brain networks during resting state. So this means that we will come across the term functional connectivity, which is nothing more than statistical temporal correlations between distant brain regions. So we can see here in the bold signal, which is what is measured mainly by the functional MRI across time, how the time series of the bold signal from different regions, distance regions, show fluctuations in synchrony across time. So in that case, we say that these areas are functionally connected and they comprise a type of network. This is the most famous network in cognitive neuroscience, the default mode network. For those that you know, it's one of the first ones that came up. But now we know that actually the brain during rest self-organizes in more such systems. Systems that they show long-range connectivity. So we have here the default mode network. Here we have frontoparietal areas from the left and the right. We have networks like that they support the detection of Cialin's important stimuli in the environment, like the detection of pain. And we also have sensory networks like motor regions, auditory areas. We see how the visual system is really decomposed in subnetworks as it's very, very complex, as well as the cerebellum. By using this systems level approach, how can we use this one to separate single patients? Actually, that was a big problem because, as we said, like sometimes patients they show different phenotypes, and we can sometimes confuse a patient that is in a low level state with a higher level state with lower, because patients can suffer from aphasia, or they cannot like they don't understand the command, so we cannot estimate high brain function. In that case, we use machine learning. As Yvan said before, he showed several methods. And here we show how we use supervised machine learning. And very, very simple terms, it's like, this is a method like when we have raw data of different type, we use a supervisor, which will label this data as this or other category, right? And then the algorithm will train to separate those labels. So when we have a new evidence, new observations, we give it to the algorithm, and it will most probably give us an answer that goes closer to this class. So we use this supervised machine learning in order to try separate patients in different classes, consciousness classes. This is a very, one of our first studies that we did, where we decomposed the brain networks using independent component analysis, and we first identified them in networks using templates. So this is a template matching. These are the templates of each network and how it fits the decomposition that we use using independent component analysis. Then we assign each network to a map, and we go for another step where we try to separate those networks that they show neuronal activity or non-neuronal activity based on some characteristics of the spatial and temporal characteristics of the independent components. And this was the first use of artificial intelligence to that matter. What we found is that this neuronality feature, when we use as a way to separate, it was informative, because we found that the neuronal components, they were quite preserved in healthy people, and there were less and less, fewer of them as we move to minimally conscious, unresponsive, and comatose state patients. When though we used this feature as a way to separate patient groups, we failed in that the performance was very, very low. And the performance we achieved, the highest performance we achieved, was to separate healthy from patients, from healthy controls from patients. So this gave us an accuracy of almost 85%. Which means that we're still left with the question, how can we use this systems level approach? So easy, so simple in nature to separate single patients. We proposed to increase our statistical power by including more patients. That was a collaborative work with the MIT. So in that case, we used as many patients as we could, and instead of using independent component analysis, we used a hypothesis-driven approach. This means we used different seeds in the brain that represent each network, as we can see here. So what we did is, like, we tried to sort which network could separate accurately patients from minimally conscious state patients from vegetative, and we found like it was a high accuracy for the so-called auditory network. Auditory network, when I say it just comes with an name due to the literature, but if we see it closely, it encompasses areas in the temporal and the occipital cortex that, if we are to identify it sensorially, this is regions that support audition and vision. And this connectivity between these cross-modalities, we found that it was very powerful in identifying and separating new patients that we have not seen before into these two classes. So we had the chance to collaborate also with colleagues from Austria, Cornell in the US, and Canada, that they shared some data with us. So we see here the training set. So this is the set that the algorithm was trained on in circles, and the test set, the new data here in asterisks. And we decomposed the feature space into 3 dimensions, like we got information from the occipital cortex and the right and the left auditory cortices. So based on this, we try to find the best separation. This is a hyperplane here of how these 2 classes can be best separated. And we found that out of the 22 new patients, the algorithm was able to classify 20, which means that this type of cross-modal interactions, a multi-sensory integration, is significant to support and separate conscious individuals. Using another type of machine learning, actually, yeah, we had, the question was like, how beyond specific hypothesis, are there brain configurations that can support consciousness more than others? And in that case, we went to see how the connectivity dynamics can help us to this matter. So this means like, here we can see this is the functional connectome and how it changes across time. So here every line, every column is a brain region. And this is a correlation matrix from negative to positive correlations. And we see how variant these cross-talking between regions it can be in conscious conditions. So in that case, machine learning was used to identify patterns, structures across the brain data. And this is just an illustration, very simple illustration of how it works. We give a lot of data to the algorithm, and then we ask to find the best pattern based on some number of clustering, for example, here 3. And by an iterative process, it tries to find new means of the data that they are less distant from one another. By doing that, we found that actually across time, our brains can show different configurations. They can show configurations that are very, very complex. As we see here, each network across this network, it can it's characterized by positive and negative connections at the shore, at the neighboring nodes, but also distant, which comes in sharp contrast with another pattern where regions really don't communicate with one another. So we can see here that the coherence between the regions is around 0. And then we have intermediate patterns that one is driven by especially the occipital connectivity, and another one that shows all-to-all, like positive connectivity. All areas talk to each other in the same way. We then went to quantify how much the occurrence rate of each pattern in different groups. So we see here the healthy people, minimally conscious state in unresponsive patients, and how pattern 1, this complex pattern, has more chances to appear in healthy situations, and how this very, very simple pattern has more chances to appear in situations where consciousness is favorable. We had also a great chance to collaborate with the Necker Children's Hospital in Paris. And they had resting state data, and they asked us to try and make sense of what they mean for hemispherotomy patients. So we had the chance to check the data of 2 patients, pediatric patients. We see here that they had right hemispherotomy, so it was complete hemispherotomy, including also the thalamus. And we can see how the interhemispheric connectivity, like how correlations between hemispheres look in healthy children, as we see here, and how this connectivity in patients would be very, very low. This is the patient MA and JJ. The 2 hemispheres will not communicate as effectively as in healthy people. Interestingly, when we looked how networks look like in these patients, we see again the sixth one, DMN, photoparietal salience, auditory, motor, and visual. And we can see how these are coming now lateralized. So on the isolated right hemisphere, now the connectivity is absent. So there is no interhemispheric transfer. Although on the other side, the networks look quite preserved. And by the way, these patients are healthy in the set, functional. They go to school, they acquire new language, they are fully integrated socially. And the question is like how they can do that with one hemisphere, and importantly, what happens to the isolated hemisphere? This was actually, it was a pertaining question, because in simple terms, it's like, can we entertain some type of consciousness in an isolated brain tissue that we don't use for functional reasons? So what we did, we used the classifier we identified in patients, as I showed you before, in minimally conscious and unresponsive. And now we used, we tested the brain of the patient MA, and this was because this patient was closer to the age group of the disorders of consciousness patients. So we see at this side the classification toward unresponsive, unconscious state. On this side, the classification toward minimally conscious and higher consciousness. And the connectivity between the temporal and the occipital cluster of patient MA was closer to the class of unresponsive patients for the right hemisphere, the isolated, whereas for the preserved left hemisphere was most closely among the class of the minimally conscious patients. Just to conclude, we used the artificial intelligence machine learning methods in consciousness research, neurology, and neuroimaging. And by this way, we managed to unravel the complexity of consciousness states. We had the chance to find single patient separation features, for that matter. And in that case, we also had the opportunity to illuminate relationships between structural and functional connectivity. And now the next challenge for us is how we can bridge different levels. So we are going from the computational level that we operate so far to the neural microcircuitry, meaning like how the networks emerge, how they're regulated at the neuronal level. I know it sounds reductionistic, but at the same time, I think crossing these different levels, we can have more insights about how the brain works, not only in disease, but also in healthy situations to inform about typical conscious states. And that would be it. Thank you so much for the invitation. I'm looking forward to discussions, and there we are.
Yes. Athena, thank you so much. And there is a first question from Professor Jean-Luc Baliban. You're on mute. Yes.
Yes, thank you, Vanessa. Athena, this is a really very nice illustration. I'm just intrigued by the very last sentence that you said, and I'd be very interested to learn a little bit more how you want to approach this neuronal circuitry. Would you go to reductionist models? In that case, which, and how would you apply some of your methods to really identify this at the neuronal level?
So at the moment, we're trying to understand how. I'm sorry, I have some lag in the image. Can you hear me properly?
Yes, we can, yes.
Okay. So at the moment, we're trying to understand how the large-scale networks emerge in the first place, trying to understand the let's say the biological underpinnings of the Bolt signal in the first case, and how this formation can work into understanding how the large-scale brain networks appear. So we go toward the hypothesis that it has to do a lot with neuronal inhibition as a biological mechanism. And with colleagues from the computational neuroscience modeling community, we're thinking that still it's a hypothesis, right? By how we can use these biological mechanisms in simulation models, trying to figure out how these can configure networks at the computational level. So at the moment, it's quite, we need to work with simulations. I hope one day we can be more invasive in order to help patients, maybe with animal models at the beginning, later with more invasive ways. But this is one way that we're thinking of doing so.
Thank you. And obviously you're focusing your research on states of consciousness, but one could even think about applying some of your approaches to states of dementia. And I'm sure that some people at your institute may already be applying some of your analytic methods to dementia and other states of altered.
Absolutely. Actually, I'm interested in different conditions where the phenomenology of conscious experience can change. Dementia can be one such condition. We have, for example, trying to quantify the how can you say, the wandering mind, how people would say that I'm thinking of this and that in healthy aging. So we have this hypothesis about mind blanking. You know, like we have sometimes the impression that you don't think of something, contentless thinking, contentless thought. And we think that this is very well supported by a specific brain configuration that we are now having good impression of what happens, but I'm not going to disclose anything because it's in preparation. I want to be sure before saying anything. But indeed, aging, dementia, situations that lead to distorted phenomenology at some point interest me deeply. And machine learning especially has helped me a lot to that matter.
Thank you.
Athena, the next question will come from Professor Benoît MACQ.
Yes, your favorite modality seems to be the functional MRI, when you observe both signals. In my teams, there are some people working on the white matter by using diffusion-weighted MRI and looking to tensor and to microstructures and so forth. When you are speaking about connectivity, do you see some way to combine the two modalities and to search for signals showing the connectivity that you observe by the functional imaging? What are the prospects to combine fMRI with diffusion-weighted MRI?
Absolutely. In the Science Advances paper where we show the different brain configurations, we actually combined, we checked the correlation between the functional connectome and the structural connectome. And we found that the highest correlation between the two was happening in the state where the areas don't talk effectively with one another. So it was mainly the underlying anatomy that was driving the connectivity. And now with my PhD trainee, we're trying more sophisticated methods about, for example, the alignment between structural and functional connectomes and the deviation from one another, how this can inform mental flexibility, for example, or disease categories. But indeed, one and the other can give more information. It's kind of finding, like constraining the whole brain's functional connectome into more meaningful ways. The anatomy can be one type of information, although more lately the genome, the human genome, can also be very, very good at that. So yeah, you're absolutely right when we need to combine the different modalities as well.
Ευχαριστώ.
Professor Casimir.
Yes, thank you very much for your very interesting lecture. My question is a question of clinician. Do you have assessed the sensitivity and specificity of these new complex methods of neurological computational analysis to move wider in routine clinical use?
Officially, not yet. Unofficially, we've been testing the classifier, at least when I was in La Pitié, very intensively. And when I came back, we're trying to use it in every clinical case we're having, just to have information about how it generalizes. At some point, I think, yeah, we need to go more applied and channel it in a more applied way and help further. I have not done that yet. I really wish I will. I'm thinking how, like as a young lab, I have so many things, and this is one of the bucket list that I have to check also. But I promise I will do it. I would love to do it, indeed.
Thank you very much.
Athena, your bucket list is so long that I think you will keep working till 100 years, at least.
If I have the same skin, with pleasure.
Yes, I wish you to have a long life.
Thank you.
And thank you so much for this conference. Et nous l'avons compris, bien sûr, un médecin qui est aidé de l'intelligence artificielle, dans un français correct. Je mélangeais l'anglais et le français. Un médecin qui est aidé de l'intelligence artificielle est mieux armé que celui qui n'en dispose pas. Et l'inverse est vrai également. La machine a besoin, on l'a entendu dans ces conférences, des chercheurs et des cliniciens pour apprendre, pour développer ses compétences. Plus précisément, professeur Casimir, l'intelligence artificielle a besoin des réseaux d'experts pour évoluer.
Yes, of course. Our next speaker is Benoît MACQ. He is full professor at the University of Louvain, where he directs the Pixels and Interaction Lab. He is active in the field of image processing and artificial intelligence. His research is carried out in an international context involving in particular the École Polytechnique Fédérale de Lausanne, the MIT in Boston, and the McGill University in Montreal. Benoît MACQ is co-founder of the Trail Institute. He is a fellow member of the IEE and senior associate editor of the IEEE Trans on image processing. He was pro-rector of the Université Catholique de Louvain and co-founder of 12 spin-off companies. The title of his lecture is Coalitional Active Learning and Prospective Applications to Network Medicine. Professor MACQ, it's yours.
Thank you very much. Just sharing the screen. Do you see the slides? Not yet, maybe.
Not yet.
Not yet.
Here, share. Okay.
It's okay.
It's okay.
We'll
Perfect.
go
Yes.
to, yeah. So I've added a word to my title, which is Continual Coalition Active Learning. So my claim and the claim of this talk is that we need new machine learning architecture. So in the past, the idea of machine learning was to collect a lot of examples. For example, on this image, there is a microcalcification in a breast. So you show a lot of image to the machine. And then after a while, the machine is learning, as it was explained by Yvan. And once the algorithm has learned enough, we use it for diagnosis, or we use it in a medical situation. My claim is that the model has to be updated continually, and that the model has to be handled not by one medical doctor, but by a coalition of medical doctors who have to share data and to share expertise to give this continual learning. Another claim in the talk is the claim of active learning. So the idea is that each annotation, each case, each data have not the same value. Some data are more informative than other data. Some annotation are more informative than other data, and the expert annotation is something very expensive. So we should ask the expert only when it is very worthy for the machine learning system. So that's the idea. Coalition, continual, and active. Active means that we will ask the medical doctor only when the annotation is bringing sufficient value to the model. So the old question was, will the AI ever one day replace the radiologist? In my field, I'm working in the field of medical imaging, but it applies also to other domain of medicine. And the answer, the old answer is, for example, Curtis Langloss in the RSNA 3 years ago said, and he demonstrated with some publication and some data that the radiologists who use the artificial intelligence will replace the radiologist who does not. So the MD who is using AI will replace the one who does not, because he goes faster, he makes less mistake, and so forth. So one of the cornerstones nowadays for the machine learning, specifically in medical imaging, but also in other fields, is the deep learning. The deep learning has really created a breakthrough in machine learning, because we reach performance that was never achieved in before deep learning. Deep learning, it's the winning in the AlphaGo, in the Go game. It's behind all the translation programs in Google, and it's of course more and more used in the field of medical imaging and in the field of medicine, in the field of radiology, in radiotherapy also we use it a lot, for example. And I will take a very simple example, which is the classification and detection of tumor in mammograms. This is a case where people think that AI could really definitely replace the MD. So how does it work, a deep learning network? Just a figure. You see there are many layers, and all those layers are taking more and more abstract signals from the image. So the idea is to take an image, to split the image into many parallel images where we search step by step. That's the why it's talk. We are speaking about deep learning. The idea is to have more and more abstraction in many layers, and it takes a lot of data to adapt the deep learning to a given situation, because we need to give a feedback. We need to feedback some data, some annotated data, to adapt all the parameters of the deep neural networks. And so deep learning, it exists nowadays, companies providing deep learning models to do breast microcalcification detection, or breast mass detection, or many detection in radiology, in screening program, or even in treatment planning. For example, if you go to a radiotherapy, the organ at risk, the tumor, at least the organ at risk are using deep learning for contouring, and it's in the practice in clinics. But so the good idea and the good way to use deep learning nowadays is to combine a whole image classifier with a local patch classifier. So the good architecture, the things on which we are working in our lab, is clearly to see the big picture globally, and to look locally, and to combine a global and local classifier in a one step. And this is the kind of architecture on which we work, and the kind of architecture that we are improving in deep learning for that kind of application. So the claim here is that the current status, it's a very recent paper which shows that artificial intelligence is, and deep learning, particularly in radiology, is doing better than one medical doctor. But if there is a second expert advice, or if there is a coalition of experts, then the AI is far behind. So the accuracy of AI is not reaching the accuracy of a double expert view on brain imaging. So that's a little bit troubling, and it seems that it's really important to keep the human in the loop, and that's what I will develop here, and how to keep the human in the loop with the less information, the less pain for the medical doctor involved in this AI-based screening. So the promising results are not reaching the accuracy that was expected. So a second hypothesis is that when you apply deep learning, so you see here in the previous paper, the model was working pretty well in some conditions. So if you are a very big hospital with a very standardized image acquisition, with experts which are well trained to annotate image, for example, by using the BIRATS annotation, if you are very strict and very well defined, you can reach a good performance. But when you deploy it in a varying situation, the model does not reach the expected accuracy. So the idea is that the model should adapt continuously to the evolution of acquisition techniques. The model should follow the guidelines, should follow the standards of image acquisition, and the model should never be a black box that is put somewhere. The idea of having continual learning is more and more in the practice of what we do for deep learning in medical imaging. The model should adapt to the guidelines. Sometimes a coalition of experts are deploying guidelines in their own way. We call that mine lines. And in some sense, the AI model, the deep learning model, should be a knowledge representation of a coalition of experts. That's what we search for. If we have a group of people with the same habits for doing screening, a group of people having the same habits to do a radiotherapy planning, how they can share their knowledge, they should have a deep learning model or an AI model which represent the knowledge of the specific coalition. So to sell a black box coming from USA and Europe or from Japan to USA is maybe not the model. We need a continual learning, and we need an adaptation of the knowledge which is represented in the model to the coalition. And so the idea is, of course, to have a model that can help for reaching consensus in a teams of medical doctors. In fact, the vision of the AI of the future is the AI being an agent which is seated in the meeting room, sitting in the meeting room with the other medical doctor, and which participate to the debate to take the final decision. This is the vision, the long-term vision we would like to push on. So I would like to elaborate a little bit more about active learning. So to make efficient model, we need annotation. We need people showing things on image, or sometimes we need, if we have cluster of interaction, we need to have some data and some annotated data. So the annotation take times and take money. And so we should try to design system where the annotation cost is reduced at maximum. And this is the idea of active learning. The idea of active learning is to provide to the learning model the most representative example, the examples that engenders the disagreement in the meeting room after, in the consensus between the medical doctor, and of course the hardest example to find. And so if we do active learning, in fact, we will ask annotation to an expert by reducing the cost of the time of the expert, but we will also provide to the expert the most interesting cases. And if we do active learning, the expert is not waiting that the AI is doing the job. The expert is interacting with the AI and keep his or her own expertise at the maximum level, because it will be challenged all the time by the AI on the hardest case or on the case where there is disagreement. So if we look the model of the active learning, on the continual active learning, we have a machine learning model, a deep learning model. We have a set of unlabeled pool, and we can use the machine learning to label all and to solve and to screen all the easy data. And when there is something on which there is a doubt, we can ask the human annotator to provide a label and to improve the machine learning inside the loop. So the human annotator, the human expert, will provide at the initial step a certain amount of labeled image, and after a while, the system will be able to ask only the important signals, the important data to the human annotator. So there are in fact three methods to be able to use active learning. The first one is to use this deep learning model with several layers and to shake the model in some sense. It's to create some randomness in the model and to see for one particular case, if I have an image to screen, an image to diagnose, if the image is entering inside the system, if I modify very slightly the parameters of the model and that I get a diagnosis which is completely different, even if the perturbation of the model on the model have been very small, then this is a case on which I have a high uncertainty. So it's also possible to use mathematical tools to measure the distance between cases and to show if a case is very different than the other case, that it is important to ask labels to the medical doctors. And the last approach to detect what are the interesting image to label is the query by committee. So the idea is to make 2, 3, 4 deep learning models which are different running in parallel. And if they do not agree, to ask the medical doctor to enter a new label. So the labels are spotting, the best label are spotting by that way. And so if we apply active learning in mammography, we have done it in my lab, you see that if there is a random choice of inputs data, the convergence of the accuracy is very slow and can sometimes never reach the best accuracy which is achievable by active learning. So you see that after 200 labels, the active learning is going to an accuracy which is far better, and the required accuracy for screening is reached only by the active learning, while it seems impossible to reach if there is a random selection of data. So the idea is not only to do active learning, but to do also coalitional active learning. Some of you are already well aware about the idea of distributed machine learning. So the idea of distributed machine learning is linked to the fact that the data, the data that have to be used are very hard to centralize. And so the idea is to have the data remaining in the hospital and to have the model moving from hospital to hospital to learn on the data. The data are remaining in the hospital and the model evolves by traveling from hospital to hospital. This is distributed learning, and sometimes people are speaking about federated learning. Federated learning is a technique which allows to preserve the privacy and which preserve the ownership on the data, which is sometimes an issue. So the idea is that for that working, distributed learning or federated learning is not enough, because we need coalition. We need people who have the same range of quality. We have the same standards of image acquisition, of working together. And so that's a first issue to solve. So we need to handle the fact that there could be a various quality of the data set. We need also the fact that we need to incentivize the people to provide annotation and to provide data. And so that's the reason why there is more and more usage of blockchain, because the idea is that to each data provided to the model, there is a token that could be claimed later if the model has some value. And we need also to protect the model. If a model is shared inside a coalition, the use of this model has to stay inside the coalition and not go outside. That's the reason why the engineers are working on a mechanism to trace the usage of the model. And so an example is a publication that my team has published 2 years ago, and we are starting more and more to deploy that kind of approach. we have a project dedicated to proton therapy. I see Serge Goldman in the room. And we do a consensus between medical doctors through this architecture of trusted coalition by using blockchain for distributed learning. So the idea of going further than a simple federated learning is to have a system when there are local data which are stored. And the idea is when somebody is providing data, he will never provide data, he will provide an increment to the model, an improvement of the model, and the improvement of the model is always validated by one or more validators. So the validators are validating the model or their own data, and they are validating also the model or on a shared set of data between the different hospitals. So when the people are providing data, they will not provide data from the hospital, they are providing increments of the model, and the increment of the model is validated on a local data set and on a global data set. And we ask each time the model is improved to validate the system by the member of the coalition. And so the old question was, will the radiologist who use AI replace the radiologist who does not? And for us, the new question is how to elaborate, how to constitute trusted coalition so that coalition of radiologists will replace the AI that does not use the medical doctor. So the old question was here, and the new one is this. So a last word to say that this kind of approach with distributed learning, with coalition learning, with active learning, we want to deploy it, and Jean-Luc Baligan has been a driver behind that, on network medicine. And there is an attempt to propose to the Région Wallonne a strategic innovative initiative around that kind of approach. And there is currently a work which is ongoing with Jean-Luc Baligan, Isabelle Salmon, Michel Georges, to deploy that kind of approach for network medicine. But I think that I have taken all my time, and I thank you for your attention. Yeah. Merci.
Merci beaucoup. Désolée, je ne parvenais pas à remettre ma vidéo. Elle était capricieuse. Alors, nous allons commencer le Q&A. Je vois qu'il y a une question directement d'Yvan SAEYS. Yvan, you can ask your question.
Thank you, Benoît. That was a wonderful talk. And it also ties into a lot of things that we're interested in. So with regard to the active learning, have you also looked into what is called weekly supervised learning models, where you actually don't require the expert to annotate very detailed, but let's say on a more broader scale, so that the expert could annotate more in a given time budget?
Yeah, that's a good point. That's one of the challenge in the Trail Institute, the Trusted AI Labs, the group of 5 universities is tackling weekly supervised things. And also weak annotation are of very big interest, because we have also some works on in the field of medical imaging related to psychiatry, and there the annotation are very vague, not very good, and we have to do transfer learning and weekly supervised learning to handle the stuff. Yes, it's a good question, and it's a very exciting emerging field, this field of weekly supervising learning, and we have young doctors starting works in this field in the Trail Consortium. Yeah. Thank you.
Next question from Jean-Luc Baligan, Professor.
Yes, thank you, Vanessa, and Benoît, thank you for this brilliant demonstration of how you can apply your mathematical tools and artificial intelligence in the medical field. I have a very naive question regarding coalition learning, because if you want to improve the AI diagnosis by submitting, difficult data to a coalition of experts to try to improve the model. How do you account for the fact that experts can make mistakes, and how do you control for that? In other words, can we even improve the quality of the diagnosis by having some external validation?
Yeah.
I mean, I'm thinking as more as a biologist than as a doctor, but
that's not at all naive. Huh? I've gone very fast on the validation of the model, but the idea. Oh, sorry, Jean-Luc.
No, go ahead.
Okay. So the one of the idea, and it's not at all naive. It's very important. It's how to validate the quality of the data annotation if we centralize the data annotation somewhere. So imagine that you have a hospital which is creating its data warehouse when there are a lot of annotated data. It's very important to have a validation of the provided data. And for example, in the field of radiotherapy and proton therapy, we use validation by seniors from juniors. The work of the juniors are validated by seniors, and we ask not only to validate, but to sometimes to rank. When we have multiple annotation on the same case, it's very difficult to say it's good or not good, but it's always more easy to see this one is number one, this one is number two, this one is number three. So to do a ranking in the quality of annotation is very good. So our idea would be to not only tokenize the annotation, but also to tokenize, to give incentive on the validation of the annotation. And we try to see how we could do that. So we know that the medical doctor needs to have points, accreditation points, and so forth. Maybe we could have one day accreditation points to contribute to an AI model and to validate annotation of juniors in a deep learning model. And that could maybe a way to incentivize people to participate to that kind of things. But maybe I'm naive on my point for this aspect of getting accreditation points by contributing to the validation of annotation in a deep learning model.
And the following question will be from George Casimir, Professor. Oh, George, we cannot hear you, Mr. President.
Okay, it's probably
Yes, better.
Thank you, Professor MACQ, for your very interesting talk. The Mayo Clinic says nobody is big enough to be alone. And I think it's a good example in your talk. But what is the qualification of the team, and how much profession must cooperate to be the most effective in the future in this field of the development of artificial intelligence in clinical use?
So it depends. If you are doing a simple screening, you don't need a lot of different experts if you are doing breast screening. But if you have complex diseases in the field of neurology or in the field of cancer, the cancer board is running in every hospital nowadays with a multidisciplinary team. And of course, the final decision is not only the decision of a deep learning model. The final decision is a consensus between the participant to the cancer board, including a representative of the patient. And so I think that the model, if the model can help and can predict what will happen for the patient, it would be a big value to increase the value of the final decision when there is a treatment plan. If we do reinforcement learning on a treatment plan, we can predict what will happen with this treatment plan. And so AI can inform better the cancer board and can inform better the deliberative process between the cancer board and the patient. And so, in fact, we have to be very modest with AI. AI has to be a tool to find new patterns, a tool to help diagnosis, but the final treatment plan has to be in the hands of the coalition. As I said, the group of people who have to take this final decision. And so that's the reason why I don't think that the approach that is followed by some startup to provide a black box to people by saying we have that such amount of accuracy in our paper and we are publishing in RSNA that we reach such an accuracy or such a precision in our treatment plan is enough. We really need to have AI which is appropriated by a team and which is transformed itself by a team. The AI has to represent the knowledge of the team and to help the team to think better. That's the point here.
Thank you very much.
Thank you, and of course you will stay with us. Thank you to all our speakers for these fascinating lectures. But we will now continue to explore these topics through the round table. Avant cela, et je le dis en français parce que ça concerne tous les médecins qui nous regardent, membres de l'ARNB ou non, cette séance a fait l'objet d'une demande d'accréditation auprès de l'INAMI. Vous connaissez l'adresse, vous la voyez s'afficher sur votre écran. Vous pouvez envoyer vos mails à contact@arnb.be. Merci de mentionner évidemment vos prénoms, noms et numéros INAMI. Professor Baligan, we will start the roundtable and the floor is yours.
Thank you, Vanessa. Well, we've heard a lot of quite fascinating new ideas and approaches, and what I would like to do is, try to make connections between the different talks and perhaps get insights from every one of the speakers, but also from the audience, some aspects that I found fascinating by comparing all the different talks. So certainly what Joe showed us is that to build networks, we first need data, obviously, and we need not only a lot of data, we need good quality data, and this may be one of the current limitations of all the data that we have in public access so far, or that we can generate locally. And a fortiori, if we consider the fact that so far what we have as available data come from either organs or multicellular preparations, whereas Yvan just highlighted the fact that we need to go to single cell, certainly not only transcriptomics, but also proteomics, and that we certainly will get more insight if we go not only cell by cell, but deeper into the cell themselves. And then from Athena, what I take out as an important message is another element that we probably do not take into account enough, and that is time. I mean, what she showed is that networks in the functioning of the brain are extremely dynamic and change over time. And you could certainly apply this to all the nodes in the networks that we build. I don't think that we need to think of networks as fixed things that are, permanently related with edges, but that the same elements of the network certainly can adopt a totally different configuration with time. And so this makes our use of data even more complex. And what I would like to discuss with the panelists is how we can move forward. What would be the key strategic orientations to move this field of network medicine into the future? This would be my first question. And then another aspect that I learned from Benoît, MACQ, is the importance of having curated data, to have a correction of the data, where human brain can certainly intervene, but in my mind, if we intervene individually, then probably we will gain some accuracy, provided the human brain is right, but we probably will lose in terms of inventiveness. We will not grasp what is unexpected. And so there's an equilibrium there that I think is also another interesting thing to discuss. And the last thing I take out from Benoît, and I think is very important, as I said initially, we need a lot of data, and one single center probably cannot handle the amount of data that is needed to have an accurate network composition. And so the use of federated learning or federated analysis is probably one of the keys. But if we come to the first question, which is how can we improve the quality of the data that we use to build networks? Let's take genetic data, for example. So far, we rely on either exome sequencing in the best of cases, but most probably and more often on transcriptomics. And again, this is taken from whole tissue and doesn't take into account the complexity that Yvan just highlighted. So this is a question to panelists and also to the participants. Where do you see the way forward?
I'm happy to start to respond, Jean-Luc, if you like.
Yes, go ahead.
Yes. I would say that from the biological perspective, the missing data are the following. First, we need some more information about time trajectories, and especially for healthy organisms. We really have no idea how normal healthy aging of a cell or an organ or a person will change with respect to the wiring diagram in the networks that we talked about, as well as will change with respect to the general as well as tissue-specific expression of the proteins that comprise those interactomes. So having a better sense of that important determinant for comparison would be crucial. Second point that I would make is that one element that wasn't discussed by any of us this morning was the metabolome. And that, I think, will also add both another layer of complexity, but also provide information about small molecular mediators of interaction between interactomes, between cells and organs. There is some data that recently has been published that attempts to correlate the metabolome with the proteome with respect to the interaction networks, and it's provided some novel and unique insights. So that's one point. Second point I would make is that as I had responded to Dr. Knopp, that there are networks of networks. In fact, that's a subfield of network science that span the different omic levels. And among those that we did not cover include the epigenome, which also influences the degree of tissue-specific expression of some of the proteins. So that complex series of networks adds another level of complexity, but also another level of interest and excitement about regulatory features that we should learn more about. I also wanted to make a comment with respect to single-cell RNA-seq and proteomics, and now even single-cell metabolomics, which is surfacing as well. I think this is very exciting, but the question then remains, should we focus on mean field effects or very specific individual elements in that complex collection of cells within an organ? And how much more informative is it to know about the profile of each cell versus the primary drivers or the median driver of a functional or phenotypic effect? Now, it may very well be that a very small number of cells in a population, say stem cell precursors, have dramatic effects on the consequences of perturbation, and we need to sort those out. We need to weight the effects of each single cell feature that we study with respect to the phenotype of interest. So I'm not, while I completely buy into the importance of doing single cell analysis, as yet uncertain, or let's say agnostic, about how one can best apply that information. All, I think, very important areas for further investigation.
Yeah, probably on this last point, and probably Yvan can also share his thoughts on this, but these single cell approaches, as you say, are very difficult to relate to the global phenotype, and especially when you're talking about the phenotype of a complex disease. But perhaps the accuracy of single cell behavior can be more useful when you go to more reductionist models, such as organoids, as he said, to try and model the cell-cell communication. I think that's what you've tried to highlight, Yvan, right?
Yes. So I think there are different possibilities of getting more mechanistic insights, also finding cell types indicative of disease states. And maybe regarding your initial question, Jean-Luc, regarding the quality of the data, I think, yeah, this is still often kind of, yeah, underappreciated. And when you're doing all these machine learning and AI approaches, actually a lot of the time goes into the pre-processing and the quality control of the data. And in our experience, these are actually, yeah, main limiting factors to the application machine methods afterwards. So what would be kind of my ideal vision for the future is that we start doing multi-center cohort studies where everybody has standard operating procedures to collect the data in exactly the same ways, measures things in the same ways, and then we will get to larger and more useful data sets because we see a lot that things cannot be replicated from just a single cohort or from a single lab, and we need to really go to multi-centric studies to have more generalizable machine learning methods that work really in different hospital settings.
Absolutely, this is an important point. Having standardized data, and especially when you talk about symptoms, you know, phenotype of complex diseases, we need to categorize all these symptoms in a very standardized way and accurate, with the accurate precision. And as you say, to gather the amount of data, curated data that is needed to have meaningful networks, probably we need to go to some federation of centers that adhere to these common standards. I think that's the idea behind the Network Medicine Alliance, Joe, right? Maybe you can, you know, explain in a few words to the audience what this initiative provides to the field.
Yes, sure. Yes. The Alliance includes multiple institutions in the EU and the US, and our goal is to create a unified approach to these kinds of questions: how best to curate data, how best to share data. We too are working on strategies for optimizing anonymity through federated approaches of the sort that Benoît talked about here, and that our hope is that by the free flow of information across these institutions, that we will gather increasingly rich information that can be useful to address some of the issues that you raised, Jean-Luc.
Would like to make comments, so please, maybe Athena first, or Benoît, sorry.
So just about your question about quality of data, I have a question to Professor LOSCALZO, to Joe. In fact, when we do imaging of tumors, we try to deploy new modalities, and we try to model the cells, and we have a proliferation model and a model of the cell's response to radiation. And in our research, what we attempt to do is to validate the model going not in the omics, but just think at the level of the cell. We try to validate the model by having a computational model of the cellular behavior inside the tumor. And when we deliver radiation, we look 2 weeks after if the model is valid or not. And so I think that one way to benchmark the quality of the data or the quality of the process could be in a very restricted case to try things and try if the model of the response is correct or not. And I would like to ask to Joe, if in the field of drug repurposing, when you do chemotherapy, there are a lot of different drugs that are available nowadays. And if we could validate a model of network medicine dynamically by looking to the response of different set of drugs on the tumor, so that we can validate the model by observing if what we predicted was really the proper prediction. It's maybe a way to validate the quality of the approach, the quality of the models, and the quality of the data. Just a question to our guest.
So that's a wonderful comment, Benoît. I think it also builds on one of the questions that Jean-Luc asked me at the end of my talk about how drugs might also be used to inform the behavior of some of the networks. And given the fact that drugs have so many targets, this makes perfectly good sense. And I'll give you a real world example of what you suggest. We have a paper now that's in revision in which we looked at the Broad Institute's drug repurposing library, which has about 6,700 drugs. All of them were put through the high throughput screen with both the wild type as well as the beta and delta variants of SARS-CoV-2. And among those that were most promising, they clearly affected not just viral entry, but also viral entry plus replication, and in a couple of cases, also processing before replication. Single drugs having those effects. Now, whether that's a consequence of the drug having more than one target or a consequence of a perturbation at a different distance through the network that the drug caused when it affected a single target, we haven't sorted out. But it precisely, I think, gets at your point that trying to understand whether the network-based predictions do in fact are valid. And if they're not, what information can we learn about the network's dynamic behavior, not just its static behavior, which is a topic I didn't have time to talk about because it's yet another rich set of questions and discussions.
I think Athena had a comment or a question. Please, Athena.
It was mostly a comment, like answering the question about the next steps in network medicine, where we can go on. And I was thinking, like, building on at least the last message I gave, that to try bridge between levels. And I was thinking, like, try bridging between disciplines even. Because, I mean, nature doesn't know the difference between genome and behavioral neuroscience, for example. I mean, I have in mind, like, this liaison across disciplines, for example, between gene expression, brain dynamics, and behavioral phenotyping. All these, they are studied with network and graph theory. They have been quantified as such. Like, and I'm thinking, like, if we combine all these different informations that they are used with, analyzed with network science, will we get new insights about prediction of diseases, or even, prevent or classification of diseases? I would even put sociology there, like really macro scale dimensions, because they contribute to disease expression in any case. So I was thinking something like that, like even vision programs that integrate different disciplines. We're going toward that. I mean, Zika is doing that. It's biomedical sciences. It embraces different disciplines. It's not just neuroscience that I used to do only. And now I start appreciating the liaison. Of course, we need the language to communicate. It's like eternal educational mode that we need to find ourselves. Like, I cannot imagine the future scientists, how broad and specific they are supposed to be asked to be. But this is kind of brainstorming I would have toward the future.
Yeah, thank you, Athena. I think this is a very important comment that we need to integrate the bigger picture, and especially the environmental component of diseases. We briefly alluded to epigenetics. Joe mentioned that certainly this is another layer of complexity, but it has to be taken into account. And obviously all the environmental influences will affect not only the networks, but also the disease. So this can be summarized as, some people talk about the exposome being one other dimension. Obviously, as you say, to collect and integrate that data, we need to not only be able to collate them, but also in a standard way that can be integrated into the analysis. This speaks to another thing that perhaps is worth discussing, which is the access to data that are in the public domain, because obviously there are many registries, either public or private, where lots of data are collected, not in the same format, but some of which could be available, and it addresses the question of interoperability between the different systems that where these data are collected. And perhaps, Benoît, you have some experience with the interoperability of different information technology systems that would allow you to collect data from different sources. I'm sure there are developments in that field, and certainly this would add even more data and make our networks probably more accurate and closer to the real life. Do you have any experience with that, Benoît?
Yes, in the field of cancer, with my friend Philippe Lambin, we try to do things for having a nice, appropriated, federated learning between entities. But Athena knows probably the field better than I do. We started with Philippe de Timery some works on the basis of the UK Biobank to get a lot of information for detecting dependencies in patients, and to have access to UK Biobank was really a plus in the research that we are starting there. But maybe, Athena, you are used to connect to this kind of large initiative. And there is also the ABCD biobank, which is very interesting for the kind of things we do in neurology.
I know of the UK Biobank. I have not used it directly, but I used other repositories like the Human Connectome project. Now we're contributing to the EU initiative for the Human Brain Project, where all data from all centers across Europe, they're supposed to be in one repository and describe different dimensions. And I have used specifically for the hemispherotomy study. I could not have done it without having access to a data set of pediatric populations that we were not able to acquire. And it was given so generously. I mean, then I appreciated all these initiatives. People that they acquired these data from the other side of the planet, now they helped us solve a problem very, very quickly and very cheap. So especially now in the artificial intelligence area, that we need a lot of data. This is the best place to get them. And this should be open. This should be accessible to scientists. I cannot imagine having to pay. It goes against this the work ethic of science to me.
Another repository of data is the databases on drugs and their targets. Joe, you briefly alluded to the Broad Institute library of molecules that you are currently screening in one of your initiatives. I suppose that this is publicly and freely accessible. I wonder to what extent additional databases, perhaps coming from the industry, would be accessible to. Obviously, if we talk about drug repurposing, there may be less interest from the industry, but maybe I'm wrong. Do you have any experience with approaching some private companies to try and share databases and knowledge there?
Yeah, that's the one forbidden zone, because as you can imagine, most pharma companies have as their one major asset their private set of drugs. That is their fundamental IP. So they certainly do share limited amounts of information if one's working on a project or has an hypothesis to test. But we rely on publicly accessible data sets. The drug bank is useful in that regard. It does list all of the at least identified drug targets through experiments for each approved drug. SIDER, S-I-D-E-R, is an interesting data set which lists all of the side effects of every approved drug, and when understood, the mechanisms. The Broad Library is a publicly accessible library, and in fact, it's accessible both in terms of chemical structures and specific chemical features of each compound, but also they will provide some of the compounds to you. And that pool of compounds was created in partnership with industry. But again, these are all compounds that have been in patients and are approved, or have been in patients in clinical trials and not yet approved.
Thank you, John LOSCALZO. Professor Baligon, we still
yes,
have,
Vanessa.
we still have
Do you have any other?
Professor Casimir has raised his hand.
Yes, Professor Baligon has considered epigenetics, but there is also a big topic, the microbiota, that represent more DNA than for one individual than his own DNA. What about the microbiota in this problem?
I'm not sure who wants to tackle that question. I'm certainly not a specialist, but it's an interesting comment, and I wonder if there are initiatives to try and integrate this additional brain, as it's called, that we have in our guts. So I don't know if any of the panelists has any insight into integration of data from the microbiome.
We're actually, in fact, looking at the microbiome's role in metabolism in patients with different cancers. And there is work that actually began at Cleveland Clinic looking at dietary molecules that are metabolized by the microbiome, and then those metabolites absorbed by the host, which, as you know, Jean-Luc, have an effect on atherosclerosis, for example. And using that kind of approach, looking at the integration of microbial metabolism and host metabolism, one can identify novel drug targets in which one drugs the enzymes responsible for conversion of food molecules in the gut within the microbiome. So you're treating the bugs and not the host. And Stan Hazen has come up with approaches for that kind of treatment to, at least in animal models, limit atherosclerosis that can be stimulated by trimethylamine oxide, which is a liver metabolite of the bacterial product of choline metabolism, trimethylamine. So that's a very, Professor Casimir, that's a very insightful question, and it's another wonderful level of complexity in this remarkable system of networks.
makes you worried about what you could do with an antibiotic treatment.
Yeah.
How you could affect all these delicate equilibrium between the gut and the rest of the organism. Right. I don't know, Vanessa, if I just want to leave some time for questions from the audience, if there are.
Yes, sure. Yeah, we made I promised something at the beginning of the session, and so I will get back with my with the question of Olivier Ferron, Professor Ferron, who will not be able to ask his question himself because he has a problem with his connection at home. So the question is, and he says, it may be naive, but are there initiatives to use protein-protein interactome, so PPI, determined based on in vivo experiments, I mean real-life setting? And that was for Joe LOSCALZO.
Yes, it's not a naive question at all. It's an important question.
No, we have very clever people watching our sessions, you know.
There are efforts being made to do what you suggest in animals, not yet in patients. And part of the reason for that is getting access to certain tissues that we can't easily biopsy in healthy patients. In the case of cancers, however, it's a different story. And we, for example, are involved in a clinical trial now of patients with non-small cell lung cancer, in which we're looking at gene expression in the tumor and comparing that to gene expression in the normal healthy lung that's removed when the tumor is removed, and mapping those differentially expressed genes to a PPI. So we're hoping to be able to create these individual reticulotypes for those lung cancer patients that might be predictive of outcome and or give insight into specific therapies that might benefit certain of them.
Thank you, Professor LOSCALZO. Professor Baligon, I think if there is nothing much to add, that we can close this roundtable, which was actually longer than what we previously planned, but very interesting. So would you like to add something, Professor Baligon?
No, I think I'm again very grateful to all the speakers and panelists for excellent talks and very fruitful discussion. I'll leave the final word to our president.
Professor Casimir, the floor is yours. Goodbye, everyone.
Yes, as we have been able to realize the way of analyzing disease in the process of changing profoundly through the molecular evolution of the mechanisms and particularly innovative aspect of biostatistics. This also leads to a much more individual medicine, both in terms of diagnosis and treatment. We heard today the pioneers of this new activity, and we have to thank them to participate to this meeting, and we have also to thank Professor Baligon for the organization of this wonderful meeting. Je vais reprendre maintenant quelques mots en français. D'abord pour vous rappeler encore le 22 octobre, vendredi après-midi, la réunion organisée par la présidente du Conseil supérieur de l'intégration scientifique, le professeur Balerio. Je crois que c'est très important que notre académie ainsi joue un rôle tout à fait majeur dans ce dossier, et le professeur Balerio l'a remarquablement préparé. Et d'autre part, notre prochaine séance qui aura lieu le lendemain, le samedi 23 octobre, où nous aurons en fait un hommage au professeur Alain de Wever qui nous a malheureusement quittés, par le professeur Jean-Louis Van Herweghem qui était quelqu'un qui a été très proche de lui. Et puis nous aurons deux exposés tout à fait intéressants, l'un par le général Nerings, un accident biologique étrange en Belgique, sommes-nous prêts? Sujet évidemment fort important quand on a vu ce qu'a donné cette pandémie du Covid-19 auquel nous n'étions assurément pas préparés. Et d'autre part, un exposé tout aussi intéressant dans un tout autre domaine par le professeur Élie Kogan, lutter contre les pratiques de soins déviantes, quelle pédagogie pour le public. Voilà, je pense, une très belle séance aussi auquel nous participerons donc le 23 octobre. Il me reste alors à remercier tout le monde, y compris évidemment tous les participants de l'audience et toutes les personnes qui ont participé à l'organisation administrative, Vanessa, bien entendu, M. Buchet, de cette réunion et vous souhaiter un très bon week-end.